Okay, I guess we'll get started. I hope everyone is here for Introduction to Computational Biology. Is that correct?
Yep.
Okay. So I wanted to give a few notes. First, there are some handouts. There should be some copies circulating. If you don't have them, you can get them at the front of the room. Beyond that, I do want to point out right at the beginning, I'm doing an attempt at course recording this year. The idea is that we'll put recordings of the lectures online. Hopefully this will be helpful to you guys. Because of that, I just ask you guys to be aware there is a camera there. If anyone has a problem with that, feel free to let me know. Please be careful about jostling the camera or anything like that, or making too much noise if you're sitting near it. It will obviously pick up you guys sitting close to it much more clearly than it will pick up me. But I hope this is something that will be helpful to you guys. We attempted this last year, and at least for the first half of the class, it was not too successful. We didn't get many good recordings. I'm trying again. The recordings from last year are online. Some are usable, some are not, and we'll try a different platform for this year. All right, before I get started, I just want to go through some administrative material for the class, and then we'll get to the real material that we'll be learning over the course of this term. But just to review, this is Introduction to Computational Biology. There are a number of different course numbers for this, depending on what version you're taking. I'll explain in detail what all of those are. But if you're here today, you're probably taking one of these four numbers. So this class this year is going to have 3 instructors and 2 TAs. So the lecturers for the first half of the class, which is the 02 or 03 251 half, is primarily going to be me, and for the second half, primarily Robert Murphy. And we'll also have an extra lecturer, Joel McManus, who will be doing some guest lectures and one module in each half of the class. They're trying to bring in some new kinds of methods that we haven't had the past couple of years. There will be 2 TAs, and I believe neither of them is here yet today. You'll probably be meeting your TAs on Friday. So each of you will be assigned to a recitation, and that will either be run by Charlotte Darby or Kelvin Liu, depending on which recitation you're in. So this class is designed in effectively 2 halves. You can take either of the 2 halves as its own class, although most of you, I believe, are taking the full class. The full class is either 03 or 02 250, and then the halves of that class are either 251, first half, where I'm the primary lecturer, Introduction to Computational Molecular Biology, which is primarily focused on topics in genomics and systems biology. And then the second half, taught by Dr. Murphy primarily, is Introduction to Computational Cell Biology, which primarily focuses on topics in mathematical modeling, various kinds of models used in biology, as well as image informatics. And I'll say more in detail about that shortly. And so the question often comes up with this class, is this the right class for you? So I'll just say that this class was designed primarily to serve undergraduate biology students. I don't think it has to be undergraduates. I think this is something primarily for biologists of any form who want to learn more about computational biology. I know as of, I guess, a year ago, it's now a requirement for the biological sciences bachelor's degree, so I'm sure that's why many of you are here today. But we think this is something that is really going to be very helpful to anyone doing experimental biology work, and that's the primary population for which it was designed. It's also designed for anyone planning to do work in experimental biology from other directions, so biology grad students, biomedical engineers, and so forth, as well as computer science students who are interested in getting more into computational biology. So I believe a number of you are planning to minor in computational biology from computer science, and that's another population for which this is designed. But basically, anyone who wants to know more about computational biology from the perspective of how it helps a biologist in biology work, this is the class we think you should be taking. Any questions about that? All right, so to go into the administrative details, this is primarily going to be written down on the handout, so if you refer to those, most of what I'll be saying in the next few minutes, you'll have a copy of it. The handouts will always be on Blackboard, but some of these you can get in class as well. So the overall structure of the class is illustrated by this diagram here. So the class is going to have 3 lectures, Monday, Wednesday, and Friday. All of you are expected to attend the 3 lectures each week. For the most part, I, at least in the first half of this class, organize it in 1-week modules, so each week is covering a different topic of computational biology. And usually the first couple of lectures are talking about the biotechnology and biology background, the computational concepts more from a theoretical point of view, and then the third lecture getting into more of the hands-on issues of how you would actually use tools and databases to work with these concepts. And then we have recitations, and each of you would take one of these 4 recitations. And they're formally called recitations, but really they're computer lab sessions, so they're the lab component of this class. Because of that, it's important that each of you goes to all of your recitations. And for the most part, the recitations are equivalent to one another. The one exception is this recitation that's highlighted in red here. So the first 3 recitations are the 03 recitations. The 4th one is the 02 recitation. So 03 is biological sciences, 02 is computational biology. And the reason we have this split is that we're trying to have one recitation that's more focused on people who have a prior programming background and can try some more advanced kinds of things we're not covering in the other recitations so much. You'll all have access to the material, so you can find out what's being covered in the other recitations. It won't affect your grade at all, depending on which you're in. We just want to give some extra enrichments of some different kinds of problems to those of you who are prepared to do some basic programming assignments in the recitations. Otherwise, it really doesn't matter too much which one of these you're in. We often get the question that someone wants to switch which recitation they're in. Usually that's fine as long as the clusters aren't over capacity. So there's a finite number of computers in the clusters where these are taught, so you can't have more people in the cluster than there are computers. But beyond that, as long as you talk to me and or the TA, it's usually okay if you switch between recitations. All right, so in terms of the course material, as I mentioned, the class is split into 2 pieces. For the most part, we have 1-week modules of the material. In the first half of this class, the 251 half, we'll be starting this week with the topic of sequences and sequence databases, which is one of the big kinds of things we work with in computational biology and one of the major kinds of data we'll be looking at in this class. We'll then move on to sequence searching and alignment, so how we would find a sequence that's related to some other sequence we know about. Protein structure and domains, so getting into working with 3D structural information, other kinds of structural information. Genome sequencing and annotation, so how we find and work with entire genomes, something that's really been dramatically changing how a lot of biology practice has worked the past few years. Genetic variations in disease, so how changes in a genome can affect phenotypes, especially with respect to diseases. Gene and protein expression, so this refers to activity of genes and proteins and the ways we study that, both biotechnology and computational tools and why we do that. And finally, what we call networks and pathways, which is often referred to as systems biology. We'll spend a week on that, and probably 1 extra lecture on phylogenetics, and then we get into either final exam if you're in the 251 number, or midterm if you're in the 250 numbers. And then in the second half, we'll cover modeling and image informatics. So basically, each of these weeks is more or less focusing on a different class of mathematical modeling that's used widely in biology. So population dynamics, biochemical models, neuronal network models. There's some material on a topic called machine learning, a very important kind of computer science for a lot of modern biology work. And then that gets into image analysis, and you'll spend the last couple of weeks on studies of techniques for analyzing biological images, and then we get into exam week. Are there any questions at this point? All right, so the homework policy, it is important to know about this, to be aware of what our policies are. So we are, I think, fairly strict on this. So everyone is expected to attend recitation sections, and those are graded. So generally, the grade is going to be either you're there and you're actively participating, or you aren't. So you don't have to worry about exactly how well you're participating for the most part. As long as you're putting in a good effort and doing the problems, you get credit for that recitation, but you do have to go to them. I know there are some acceptable reasons for being absent. I'm generally not too open to being absent because you have more work to do or anything like that. But if you have to attend grad school interviews or something like that, talk to me and we can make other arrangements. Homework assignments will more or less be each week. They'll be handed out in your recitation. They'll also be on Blackboard. And they're due on paper in lecture the Friday after they're handed out. So they'll be handed out on recitation on Friday, due a week later with some exceptions for weeks where we don't have a full week of classes. So please make sure your assignment is here in class on paper at that time so you can get it to the grader to go through those. Exams will depend on whether you're in the full term or the half-term version of this. So however you work it, exams count for 40% of the grades in the class. If you're in the full-term class, then there will be a midterm that's 15% of the grade, and a final that's 25% of the grade. If you're in the half-term, it's just one final exam for the half-term that's 40% of the grade. Because of the way this works in the kind of two-half split, we'll generally have 2 exam days with different exams at each of these times. It's not the same exam, but it's equivalent. We'll scale them to be on the same scoring. So generally, we can be pretty flexible about people who have a conflict and need to switch from one to the other. So if you're scheduled to take the midterm, you can take the 251 final instead. Just talk to me in advance because we have to know how many people are taking each of these. So that's just a quick summary. And again, this is on the course information handout you have. Any questions about this? All right, so a few of the basic policies you should be aware of. We do not generally give extensions on the homework assignments. They're due the day they're due. There are some special cases, if you get in a car accident or whatever and you're in the hospital the day it's due, okay, we'll make an exception for that. But beyond that, I generally do not make exceptions for homeworks being due when they're due. You have a week to do them. I guarantee you don't need a full week to do them. So if you know you're not going to be here the day the homework is due, do it early and make arrangements to get it to me early. There may be some opportunities for extra credit. This is something that we'll really only consider if it's at the end of the term and you're right around the borderline of one grade versus another. If you're in that situation, then please talk to me or Dr. Murphy. We might be able to make arrangements. Please don't come to me after the first homework and ask for extra credit. I'm not going to be open to it at that point. Finally, our collaboration policy. Like all classes here, we take academic integrity very seriously, so please make sure you understand what is allowed and what is not allowed as far as collaboration. So we do allow and encourage you to work in study groups. Feel free to talk to each other, learn from each other, but you have to do the assignments on your own. So we list a few examples of things here that are considered unacceptable collaboration. So if you need to run analysis on the computer, you each need to go do that on your own and turn in your own results. You can't just do it once as a group and turn in shared results. You're not allowed to copy any portion of anything from one another. And if you use a method suggested by another student, it's important that you understand that and can explain how that method works and actually execute it yourself. So is this clear to everyone? Okay. So textbooks. In the first half of the class, we do have a required textbook. So this is Pevsner, Bioinformatics and Functional Genomics. I will say that I could not find any textbook yet that really covers what we want to cover in this class, so I don't follow this textbook that closely. But some of the topics, I think it really does have very useful readings that will help you understand the material of the lecture. So those are listed on the course calendar handout you have here. We also have one online textbook. This is really a reference to some of the tools and databases we'll be using. I think it's something you can kind of refer to after the fact if you have questions about them, but it's a useful thing to have access to. So again, this is free online. You can use it as you need it. We'll also give extra handouts online, so some additional readings. For the second half of the class, Dr. Murphy uses solely these handouts online for reading. For the first half of the class, for the most part, it's in the textbooks, and then it'll be on Blackboard if there are other readings beyond that. Has anyone had any trouble finding the textbook? Okay. So the next thing I want to discuss is the course objectives. So every class here at Carnegie Mellon, we put together a list of course objectives, which are what we believe you should be getting out of this class. You can, of course, review these slides online if you want to know in detail. I'm not going to go through word for word what these say, but these are the things we set up that we believe you should come out of this class knowing, and that we use to figure out whether you're learning what we want you to learn from this class. And these are the official course objectives. But I'd like to say that really there's an unofficial course objective, and that is to teach you what you need to know about computing to be the best biologist you can be. So I know a lot of you may not have had much or any exposure to computational biology or maybe any kind of computing before, and I want to be clear about what it is that we hope you will get out of this and what we expect you to be learning as you go through the term in this class. So one of the key things, of course, is to know what computational tools are available to you as a biologist and how to use these effectively. And tools is defined fairly broadly here. In some cases, it's literally a computer program that's widely used in biology that we think you should know about. Very often, it's a kind of data source, so knowing about repositories of sequence data, or structured data, and so forth, how people use those. But in other cases, it may be something more abstract. In some cases, it's an algorithm, and it's important for you to understand that algorithm. So basically the way the computer program works, if you want to be able to use that effectively, and maybe eventually develop your own programs or your own modifications to these. In some cases, it's simply a way of thinking. So a lot of what computer science and computational biology is about is really thinking about problems in a certain way. We're often thinking formally, abstractly, and mathematically about a very complicated, messy biological system. And we hope that by the end of this class, you're familiar with that kind of thinking and how it can inform your work with computational tools in biology and make you a better biologist. We want you to understand what kinds of problems these tools can help you solve. And to a large degree, this is again going to be problems that we think those of you working primarily in experimental biology will be solving, but where computational tools have become a very key part of how every biologist, experimental or not, is doing biology today. This can be thinking about how you would design an experiment, how you would evaluate a wet biology lab is going to be driven by computational ways of doing these things today, and we want you to understand how that works. To do this, you're going to need to know a fair amount of computer science. Obviously not the equivalent of taking, let's say, a whole degree in computer science, but there are some key concepts that you need to know from how a computer scientist would approach a problem. So how to take a complicated, messy biological system and reason about abstracting it, formalizing it mathematically, identifying good methods to solve it, and interpreting the answers that come out of these methods. And as we go through the term, I will be revisiting again and again how we need to think about these issues and how being able to think like a computational person will help us work more effectively with these tools. Some of you might be surprised to know, though, that a lot of what we're going to be learning in this class isn't computing, it's biology. You can't work effectively with computational biology unless you understand the biology well. And in a lot of cases, the biology we need to know is not the same biology you might learn in your other classes. So I'd say probably about a third of the material I'll be covering in the first half of this class is really material on biology, so understanding the background of the molecular biology, the biotechnology, and so forth that we need to know to use computational tools for working with and understanding that biology. Some of that's as simple as understanding what kinds of biological questions computational tools can help us solve. Often it comes down to the question of understanding data. So a lot of what we're doing in this class and a lot of what is really going on in biology today has to do with understanding and working with data. You need to know where that data comes from, what the properties of that data are if you want to do that effectively, and also understanding always how our computational abstractions that we need to develop computer algorithms and computer programs differ from the real biological system. They're always approximations, they're never going to be exactly the real system, and you have to understand the difference between these if you want to know how to use computational tools effectively. So that really is what I hope, and I think Dr. Murphy hopes you'll be getting out of this class, and what you'll be able to do, and what you'll be able to reason about by the time you get to the end of the term. So are there any questions about any of this? Okay. So that brings us to the beginning of the real technical material of this class. And what I wanted to do for the remainder of this session is start getting into material in sequences and sequencing, which is the first topic we're going to be covering in this class. So it's important that you understand what a biological sequence is, where these come from, and some of the things that are interesting to us in understanding how to use computational tools with them. And a lot of biological sequences flow from the central dogma of molecular biology. So can anyone tell me what the central dogma of molecular biology is? Is this a term you guys have heard before?
The DNA, RNA, and proteins, and then the feedback from DNA itself replicating.
Yeah, so essentially the central dogma says information flows from DNA to RNA to proteins. So from DNA through the process of transcription, and of course replication of DNA to itself, but transcription to RNA, and then translation from RNA to proteins. And each of these steps carries with it particular kinds of sequences. So we have nucleotide sequences for DNA that we will commonly represent as strings of A's, C's, G's, and T's. Again, it's always important to remember that the string of 4 letters is an abstraction. It's not quite the same thing as actual DNA, and sometimes the distinction is important. But often we need to represent things abstractly and simplified to get good computer algorithms. Nucleotides also come from RNA, strings of A, C, G, and U. We have uracil instead of thymine in RNA sequences. Usually in the databases, the U's will just be written as T's anyway, just something to be aware of. But remember, it's actually a different nucleotide in RNA. And then in proteins, we'll commonly represent these as amino acid sequences. Usually when working with them computationally, we'll use this 20-letter alphabet, so 1 letter for each amino acid. If you haven't seen that before, it's in the readings. It's good to get to the point that you remember which letter corresponds to which amino acid. Sometimes it's important to be able to recognize these. So when we get into where these sequences really come from, it can be important to know some details about how central dogma works. And there are really 2 variations on central dogma. So central dogma for prokaryotes and central dogma for eukaryotes. For most of you guys, I think this will be largely a review of things you've seen in earlier classes, but there's some aspects of this that's important for us to understand as we move on to more advanced topics. So in the prokaryotic case, normally we will have DNA contained in a circular chromosomes, and those chromosomes are organized into operons, where each operon will typically contain 1 transcriptional unit, so 1 piece of RNA that is transcribed from that operon. There will canonically be signals in the operon, so signals typically upstream of transcription start, canonically 1 at the -10 position, approximately 1 at the -35 position, and those are used by the cellular machinery to help recognize that there is an operon in this region of the genome. That results in transcription, and then there will be a piece of this DNA that is transcribed into an RNA by an RNA polymerase. There will be another kind of signal in the sequence called the terminator signal that tells the cellular machinery that it's reached the end of this operon and that it should stop transcribing RNA. Within that region, we will typically have 1 or more protein coding regions. In prokaryotes, we generally identify these by looking for what are called open reading frames or ORFs. And an open reading frame basically means a region from a start codon, usually AUG, to a stop codon, usually UAA, UAG, or UGA, in frame of the start codon. There will be additional sequences, typically in the RNA, that are helping the translational machinery recognize that there are regions to be translated here. So canonically, what's known as a Shine-Delgarno sequence, just upstream of the protein coding region. Within that, we'll then have these protein coding regions translated by ribosomes, and you should, I expect, almost all of you be familiar with the idea of a triplet codon alphabet. So triplets of nucleotides will each code for an amino acid, and then these sequences of triplets will give us proteins or polypeptide sequences. These will canonically be what we would refer to as precursor proteins. I'll come back to that in a moment. But basically, that's how we get from DNA to RNA to protein. And an important point in this is that this is a process that is guided by patterns or motifs within these sequences. The cellular machinery looks for particular patterns. It recognizes particular patterns that tell it how to operate on this particular kind of sequence. So we have these sequences upstream of the transcribed region, upstream of the operon, telling the transcriptional machinery that there is an operon here. We have sequences that help the cell, the ribosome, then identify that there is a region to be translated into a polypeptide sequence here. But it's important to note that these signals are not perfect. They're not always conserved, they're not always the same, and this will have some important consequences for us. Often these signals are just missing. So you'll see in your molecular biology textbooks that there's supposed to be a signal at the -35 position. Sometimes the signal's there, sometimes it isn't. Even when these signals are there, they're usually not exactly the consensus or average sequence. They're usually perturbed a little bit from that. So this is important in part because we can't always identify when we're looking at a piece of DNA whether or not these signals are there. We can be sure from looking at a piece of DNA and seeing a pattern of sequence whether we're actually looking at an operon. And this is very important for computational purposes because computer algorithms look for the same signals the cell do, and they don't always get this right. So just be aware there are these signals. The cell doesn't always interpret them the same way, and our computer algorithms aren't always going to interpret them correctly. The same applies in protein translation. Your molecular biology textbooks will probably say that there should be a Shine-Delgarno sequence. Really, only about half of prokaryotic polypeptide regions have a Shine-Delgarno sequence upstream of them, so sometimes it's there, sometimes it isn't. It helps, but it's not necessary. Start codons you'll usually see referred to as AUG, but it's not always AUG. You might have an AUC start a protein coding region, and if you're using a computer algorithm to predict the proteins, it may miss that if it doesn't know that sometimes you see these. In almost any organism, this can occur rarely, so some small fraction of the genes might have a non-canonical start codon. In some organisms, it's a very large fraction of the genes. So any of you who have done phage hunters might be aware that lots of phages use a very large fraction of the time a non-canonical start codon, so that's something you need to be aware of in working with those. Sometimes they will read through a stop codon to put in an amino acid instead of stopping there. Some organisms will do this rarely. Some organisms will reliably insert an amino acid instead of stopping at one of these three stop codons. So again, that's something to be aware of. Are there any questions at this point? Right, so proteins as well, it's important to know that what comes out of the ribosome is often not the final protein product. So I refer to that as a precursor protein because very often that undergoes further modifications after it comes off of the ribosome. Parts of it may be cleaved off, it may be cut in various ways, it may be modified, there may be some other kinds of chemical changes that go on in certain positions. So what's actually functional in the cell, what's called the mature protein or mature peptides, may not be the thing directly encoded in the DNA. And this is going to be another very important point because for the most part today, when we have amino acid sequences, proteins, we have these predicted from looking at DNA sequences. And it's important to know that what's functional in the cell is not necessarily exactly the thing encoded in the DNA. So putting it all together, here's just a quick summary. For prokaryotes, we have circular chromosomes. These are transcribed from the operons into RNAs. Within each operon, you may have one or more protein coding regions, and ribosomes will produce protein sequences, precursors, which may be modified in some ways to produce mature proteins. Any questions? Right, so we often want ways of quickly summarizing a lot of this information that will often be shown in what's called a gene structure diagram. These may be drawn in slightly different ways, but it's kind of a quick summary of the genes you would find within a region of a prokaryotic genome. And usually what we would do is denote some of the key pieces of information we would need to know to reconstruct where the transcribed regions and where the translated regions are within that. So this would be an example of what a gene structure diagram might look like for a prokaryotic operon, showing the position of one transcribed region and the translated regions within that. For eukaryotes, things are qualitatively similar, but there are some important details that are different. So I'll just go through now how things are different in eukaryotes. So the central dogma for eukaryotes usually starts with linear rather than circular chromosomes. These are organized into genes, and unlike in the prokaryotic case, we will typically have one protein coding region per gene. There will be signals around the start site of this. They're not quite the same signals as we see in the prokaryotic case, but they're similar sorts of signals. In the eukaryotic case, we will much more often have transcription factor binding sites that are used to regulate how the gene is active. So sites in the DNA that are recognized by proteins that may or may not be present, or may be present at different levels, under different conditions, different points in time, or different cell types. So this happens in prokaryotes as well, but it's much more extensive and much more important in how eukaryotes regulate gene activity or gene expression. So we will, however, have a transcribed region converted by an RNA polymerase into a pre-mRNA. So it'll look something like this. And in the eukaryotic case, unlike the prokaryotic case, these pre-mRNAs will typically be separated into exons and introns. So the protein coding region is contained in the exons, and the introns will be removed to produce a mature mRNA through the process of splicing. So a molecular machine called a spliceosome will come along. It will recognize other kinds of signals around intron sites. We'll figure out that there is supposed to be an intron here, cut that out, and then concatenate the exons to produce the mature mRNA, which will then typically contain a single protein coding region, so a single ORF running from a start codon to a stop codon. Upstream of that, we will typically have what's called a 5' untranslated region, or UTR, downstream a 3' UTR. These often contain other kinds of signals that are important in regulating the behavior of this RNA, such as controlling its lifetime, so how long it lasts before it's degraded in the cell. A ribosome will then come along, just as in the prokaryotic case, produce a precursor protein, and that part of it is more or less similar, conserved across all kinds of organisms. One important thing to be aware of in the eukaryotic case is that the process of splicing is often conducted different ways in different transcripts of the protein. So in other words, we have what's called alternative splicing, where a single precursor mRNA or a single pre-mRNA may be spliced in multiple different ways. Some exons might be omitted from some transcripts versus others. Sometimes the start or stop sites of the exons are different. Sometimes the whole transcription will start or stop at different positions at different times. But it's important to note that a single pre-mRNA may be spliced in different ways, and this is a very important part of regulation of genes in eukaryotic organisms. The more genetic sequences we see, the more the estimates keep going up of how much alternative splicing there is. But the current understanding is that most eukaryotic genes have a small number of splice forms, so maybe on the order of 3 or 4, but some genes can have thousands of different splice forms. Sometimes this splicing process is purely stochastic, so some fraction of the time you get one form, some fraction you get another form. In other cases, the splicing process is regulated. So it may be that in some kinds of cells you always splice this way, and other kinds you always splice this way, or at some times or under some conditions. So again, that's something to be aware of. It's also worth noting that the signals that control this are not as well understood as some of the signals controlling other kinds of processes in the cell. So when we use computer algorithms to predict what the mature RNAs are from looking at the DNA or looking at the pre-mRNAs, those predictions are not necessarily something you can trust. They might find one splice form and miss others. Later in the term, we'll be seeing how those predictions work and seeing in more detail why that's the case and how we should be using that in practice. In the eukaryotic case, we also, as in the prokaryotic case, typically have differences between the precursor protein and the mature protein. And this, again, is something that is typically more complicated and more important in eukaryotes than prokaryotes. So eukaryotic proteins are often glycosylated. They have often complicated sets of sugar groups attached to them that can change how they behave. They often have transient modifications. So a protein might have sites for methylation or phosphorylation. And it may be that sometimes these are methylated, sometimes they're not methylated, and the behavior of the protein may be different depending on whether or not that methyl group is attached. So again, it's important to know that what's encoded in the DNA is not necessarily what you see in the cell, and even what you see in the cell at one particular point in time or one cell type might not be what you see in another cell or at another point in time. Any questions about any of that? All right, so eukaryotes, we can also summarize this information in a gene structure diagram. It's worth noting that the gene structure diagram for a eukaryote looks very similar to that for a prokaryote, but usually within these transcribed regions, instead of these boxes representing different polypeptide sequences or different protein coding regions, they would represent exons within a single protein coding unit. So just be aware that a similar diagram may represent different things in these two cases. Any questions about any of that? All right, so the last thing I want to cover in the remaining 15 minutes or so of class is where sequences come from. This is going to be something we'll return to later in the term as we get to how you get relatively large sequences, so things like a whole genome. But I want to say a little about the basic technologies of sequencing, because it will be very important for us to understand where our data comes from and what kinds of errors are introduced in different kinds of data depending on where it comes from. Most methods for DNA sequencing or RNA sequencing rely on the fact that cells have evolved to be very good at replicating DNA. So DNA naturally replicates. Basically, if you have a strand of DNA, you throw in free charged nucleotides, a polymerase, and a primer, which is a little piece of RNA that helps get things started. That's basically all you need to replicate your DNA. You will produce a complementary strand, so basically aligning A's with T's, G's with C's, generating 5' to 3', complementary to your original. And this machinery was basically something that biotechnologists figured out many years ago, how to co-opt to create techniques for sequencing DNA. And the most popular method, the dominant method for most of the history of molecular biology was a technique called Sanger sequencing. And Sanger sequencing basically took this process of DNA replication and just threw in one extra piece, what we would refer to as a terminator base or a dideoxynucleotide triphosphate. And a terminator base is basically a chemically modified nucleotide that has the property that nothing else can attach after it once it's incorporated into a sequence. So if you throw in the normal 4 bases, but you also throw in a terminator T base, let's say, then it will start replicating, but every so often you'll incorporate a terminator T complementary to an A. And once that happens, the replication is stuck. That strand can't replicate any further. And this has the property that if you have lots of strands in parallel, so you've got a nicely replicated or purified piece of DNA, and you run this reaction many times in parallel, every time you have an A, you have some chance of incorporating a good T and continuing the replication, and some chance of incorporating a terminator T and blocking it. So after running many copies of this, what you'll end up with is strands that run out to all the positions where there was an A in the original strand. So strands complementary, but stopping at each of the possible A's, up to more or less a, you can do this for up to about 100 A's before you start or 100 nucleotides, maybe up to 1,000 or so before this starts breaking down. But basically what you get out of this is strands whose lengths correspond to the positions of the A's in the original sequence. And we can use this by taking a technology for measuring lengths of pieces of DNA to create a technology for sequencing. In particular, if we use a polyacrylamide gel, which lets us sort sequences by their lengths, we can run out strands corresponding to each of these polymerized pieces and see everywhere the strand got stuck. And that's going to tell us exactly where there are T's in these stuck strands, which is going to tell us exactly where there were A's reading 3' to 5' in the original strand. So we can read off where the A's were in the original strand. And if we repeat that for the other nucleotides, then we can read off where the T's were, and the C's were, and the G's were. So in other words, what we can figure out is that our newly replicated strands read GAA, TGAC, and so forth. And that's telling us that the original strand in complement read in reverse through the complementary strands TACCATA, and so forth. So in other words, that's basically how DNA sequencing worked. And originally this is something people would run manually in their laboratory. They would actually run these reactions, create a gel, run out the gel, and manually read off these bases. Eventually this process was automated, so you could put all of this in a machine that would do this entire thing automatically, read out what's called the sequence trace, which would tell you the order in which these bases are coming off the strand. And that made it possible to start doing this on very large scales. So to sequence maybe 1,000 or so bases at a time, and to do this for many strands in parallel by just cranking these machines, many machines typically, all at a time, running all day and all night. And that really is what ushered in what we would call the genomic era of biology. We could start getting large amounts of sequence, start coming up with sequences of entire organisms, or entire what are called transcriptomes, so all the RNAs in an organism. And that is something that's really dramatically changed how biology is being practiced today. And that's something that's going to underlie a lot of what we'll be seeing in the first half of this class. But that basically is Sanger sequencing. Before I go on, are there any questions about this? Right, so it's worth knowing that Sanger sequencing, maybe 10 or so years ago, people started moving beyond this, and it's, by this point, ceased to be the dominant technology for sequencing. So it was replaced by what's often referred to as next-generation sequencing. And next-generation sequencing isn't a single technology. It's a lot of different technologies that people have tried to do sequencing faster and more efficiently than Sanger sequencing. This is just illustrating one technology called pyrosequencing, where effectively what you do is do something like Sanger sequencing, but on lots of little microscopic beads that you can trap in little wells on a plate. So each of these little wells becomes like a Sanger sequencer running in parallel, and you can do many, many Sanger sequences in parallel and get a lot more sequence a lot faster. So I don't think it's so important for our purposes to know too much about the particular technologies people are using today, because this is a very active field of research. The technologies are changing all the time, and no one really knows what the dominant technology will be in 5 to 10 years. But we can be pretty sure that new technologies will come along, they will be faster, and they will be cheaper than the technologies we have today. And so more and more sequences are accumulating, and there has really been an exponential growth in the amount of sequence we have available that's been going on for decades now, and shows no signs of stopping anytime soon. For the most part, these new sequencers involve some trade-offs. They can generate large amounts of sequence much more cheaply than the older sequencers. They can generate much more in any given unit of time. So gigabases of sequence per day, so billions of bases a day versus maybe millions from the earlier technologies. They usually give us shorter pieces of DNA. So the Sanger sequences, you can get maybe 1,000 bases in a row. The newer technologies, for the most part, are giving you things that are more in the range of tens to a couple hundred bases. And this will become an important point later when we get into looking at large pieces of genome. So sometimes people do still use older or less efficient technologies if you need big pieces of genome. But basically, there are lots of these technologies available. And as we get into using the sequence information, I'll try to revisit again and again the issue that these technologies have trade-offs, often different kinds of performance, different kinds of errors or mistakes they tend to make, and this can be very important in understanding how to use and interpret the output of these. So that basically is nucleotide sequencing. I'll go through more quickly amino acid sequencing because realistically, amino acid sequencing is not nearly as important today. It's not nearly as high throughput, so you can't get amino acid sequences as efficiently as nucleotide sequences. So for the most part today, if people want to know protein sequences, they computationally predict them from DNA sequences. So it's much easier to sequence DNA or RNA than protein. The classic way to sequence a protein is what's referred to as Edmond degradation, which basically is a technology for using a specialized kind of molecule called a phenoisothiocyanate, or a PTC, which basically has the property that it can grab onto one amino acid, so the N-terminal amino acid of a protein sequence. So if you purify your proteins, you can grab all the N-terminal amino acids. You can then pull those off of the proteins and isolate these single amino acids from the rest of the protein. It's not entirely an error-free process. There will be some mistakes, but you can more or less get these off and see that what they pulled off were, let's say, M's, methionines, and then figure out that your first amino acid was a methionine. You can then take what's left, do this again, attach more PTCs, pull off the next thing, and see that mostly you get off L's, leucine, so probably the second amino acid was a leucine. You'll see some mistakes, so sometimes the amino acid won't come off, or you might have some impurities or whatever. So you can only do this a small number of times before the mistakes start overwhelming the actual data, but you can get short sequences, sequences of peptides, maybe 10 or so amino acids, and then you can use other techniques to try to get full proteins. Typically what's done to get longer pieces of amino acid sequence is you digest the protein with protease that will cut it up into short pieces, you sequence the short pieces, and then it's kind of puzzle to figure out how to put together different sets of short sequences to create a unique long protein. So that is a pretty accurate way of figuring out your amino acid sequence. If you want to know the sequence of your mature protein, that's really the best way to do that. But it's not something that's done so much anymore, just because it's, as I said, it's not nearly as efficient as sequencing nucleotides. More often, people are using mass spectrometry. So mass spectrometry is a technology for measuring masses of molecules. This is something that people are using to try to make protein sequencing more high throughput, try to do something more equivalent to what we can do with nucleotide sequences, but really it's not there yet technologically. It's not nearly as efficient as nucleotide sequencing. The basic idea behind mass spec, though, is that we will digest our proteins into little peptides, and then we will do a secondary fragmentation to break these peptides into even smaller regions. And then we will look at the masses of these little pieces, these little subsets of a peptide. Strictly speaking, mass to charge ratios, you can more or less figure out masses from there, and try to look for what are called ladders of masses that correspond to differences in a single amino acid. This is simplifying quite a bit, but roughly speaking, if you find there's a mass that corresponds to a T, a threonine, another mass that corresponds to 2 T's, another one that's an R and 2 T's, an S and R and 2 T's, and so forth, that's a pretty good hint that probably there's a peptide that had that sequence of amino acids in it. More complicated than that, these signals are really messy. There's a lot of uncertainty that goes into this. But roughly speaking, this is how people are trying to automate the sequencing of proteins. It's pretty fast. It is something you can well automate with good computer algorithms. But there are a lot of mistakes that it makes. So it's not really something very reliable. It doesn't work so well for what's called de novo sequencing. So trying to sequence something you've never seen before, there's a pretty high chance of mistakes. It can work quite a bit better if you're trying to sequence things that are already in the database. So you can look at the peptides you discover, look through databases of these mass spectra or just sequences that are known, try to see if you're finding something that's already known. That can clean up your data quite a bit and give you a reasonable amount of accuracy. But roughly speaking, that's where sequencing technologies are today. Most sequence today comes in the form of DNA or RNA sequencing, and usually amino acid sequences are predicted today from the DNA or RNA sequences. Often these sequences are coming as parts of big sequencing projects, sequencing whole organisms or even groups of organisms, and then everything is computationally predicted from looking at this complete genome sequence or the complete transcriptome. So the transcribed sequences, RNA is sometimes predicted, sometimes directly sequenced. Usually you'll want to pay attention to which it is when you look in the databases. The databases today contain vast amounts of information. So there are huge amounts of things that a biologist working 20 years ago would have had to do themselves, that today you can just go and look up in a database. If you're working with any standard model organism, you will probably find its genome sequence in a database somewhere, often many variations on that. There are literally terabases of complete sequenced assembled genome sequence, and petabases now of pre-sequencing reads. This includes thousands of complete organisms, and the technology is constantly improving. So this is something where what we have today is probably a small fraction of what we'll have in just 5 years from now. But basically that's where things stand today. Putting back up this slide of information on the instructors, and that's one last time. Are there any questions before we break for today? Yeah. Thank you.